Biology 2 DNA RNA protein synthesis test, Form A. Number 1. The building blocks of DNA and RNA, or nucleic acids, are A. Nucleotides, B. Amino acids, C. Carbohydrates, D. Proteins. Number 2. DNA contains the sugar A. Ribose, B. Deoxyribose, C. Lactose, or D. Glucose. Number 3. Unlike DNA, RNA contains A. Thymine, B. Phosphate groups, C. Uracil, or D. Adenine. Number 4. What is produced during transcription? A. tRNA, B. mRNA, C. DNA, or D. Proteins. Number 5. If a segment of DNA contains the following sequence of nitrogenous bases, what would the sequence of RNA nitrogenous bases be following transcription? TGG, ACG, CAT, GTC. Is it A, UCC, TGC, GTU, CUG, B, ACC, TCC, GTA, CAG, D, ACC, UGC, GUA, or CAG? Use figure 12.5 to answer questions 6 through 7 below. Number 6. What process is illustrated? A. Mutation, B. Replication, C. Transcription, D. Translation. Number 7. Identify structure X. A. tRNA, B. Codon, C. mRNA, or D. Amino acids. Number 8. What happens during the process of translation? A. The ribosome uses information from proteins to create DNA. B. Transfer RNA is made from messenger RNA. C. The ribosome uses information from messenger RNA to produce polypeptides, which will soon form proteins. Or D. Messenger RNA is made from proteins. Number 9. Use the mRNA codon chart to translate the following DNA sequence: TAC, AGG, CGT. A. Methionine, leucine, or cysteine. B. Methionine, serine, alanine. C. Methionine, asparagine, isoleucine. D. Methionine, arginine, or tyrosine. Number 10. How many mRNA bases are needed to make 3 amino acids? A6, B12, C3, or D9? Number 11. A change in a sequence of DNA is called A. Mutation, B. Polygenetic trait, C. Recombination, or D. Single gene trait. Number 12. Which of the following is or are a frame shift mutation? Select all that apply. A. Deletion, B. Insertion, or C. Substitution. Number 13. Each set of 3 nitrogen bases on mRNA that codes for an amino acid is referred to as an A. Anticodon, B. Base pair, C. Codon, or D. Nucleotide. Number 14. Blank carries amino acids to the ribosomes for the assembly of proteins. A. mRNA, B. DNA, C. rRNA, or D, tRNA. Number 15. What process is shown in the figure below? A, DNA replication, B, translation, C, transcription. 16. Identify the part of the nucleotide labeled X in the diagram to the right. A, sugar, B, nitrogenous base, or C, phosphate group. 17. Enzymes play an important role in a lot of biological processes, especially DNA replication. Which enzyme is responsible for pairing nitrogenous bases together to create a complementary strand of DNA? Is it A, the helicase, B, DNA ligase, or C, DNA polymerase? Number 18. Identify the part of the nucleotide labeled Z in the diagram to the right. A, nitrogenous base, B, sugar, C, phosphate group. Number 19. Identify the type of mutation shown in the DNA sequence below. A, substitution, B, deletion, C, insertion. 20. Identify the type of mutation shown in the DNA sequence below. A, substitution, B, deletion, C, insertion. 21. Identify the type of mutation shown in the DNA sequence below. A, substitution, B, deletion, C, insertion. 22. Which of the following sequences represent frame shift mutations? Select all that apply. A, sequence number 1, B, sequence 2, C, sequence 3. 23. A change in just one DNA base makes the difference between a person having healthy red blood cells and a person having sickle cell disease. Determine the mRNA nitrogenous basis that corresponds to the DNA nitrogenous basis and the mutant hemoglobin gene. A, GUA, B, CAU, C, CAT, D, GTA. 24. A change in one DNA base makes the difference between a person having healthy red blood cells and a person having sickle cell disease. Determine the name of the amino acid that is wrongfully placed in the hemoglobin. A, the histidine, B, the alanine, C, the valine, or D, the glutamine. 25. Name the amino acid that would be called where the W is in the diagram below. A, glycine, B, serine, C, proline, or D, alanine. 26. Name the 3 amino acids that would be placed where Z points to in the figure below. A, Lea, Ala, HIS, B, LEUSER, ALA, C, PHEGLY, ALA, or D, ASP, ARG, VAL. 27. Which scientist is or are credited with building the first 3-dimensional model of the DNA molecule shown below? A, Watson and Crick, B, Rosalind Franklin, C, Hershey and Chase, or D, Erwin Chargaff. I apologize for those taking the test. The pictures will not load on the audio recording, so please refer to your paper copy. Okay, number 28. Observe the data below collected by Erwin Chargaff. Which of the following conclusion best supports the data? A, DNA replication is semi-conservative. B, DNA structure is anti-parallel. C, nitrogenous bases are held together by hydrogen bonds, or D, DNA base pairing is complementary. 29. Which is the correct sequence of the transfer of genetic information in most organisms? A, RNA to protein to DNA. B, protein to DNA to RNA. C, DNA to RNA to protein. D, RNA to DNA to protein. 30. Which of the following is a correct statement regarding transcription and translation? A, transcription occurs only in the nucleus and translation occurs in the cytoplasm. B, translation occurs in the nucleus and transcription occurs in the cytoplasm. C, transcription occurs only in the nucleus, but translation may occur in both the nucleus and cytoplasm. Or D, both transcription and translation can occur in both the nucleus and the cytoplasm. 31. How does base pairing lead to the formation of a double-stranded molecule of DNA? A, the base of one strand is connected to the nitrogen base of the complementary strand via hydrogen bonds, or B, the base of one strand is connected to the nitrogen base of the complementary strand via covalent bonds. 32. The three nucleotides highlighted in green in the diagram below are called A codon, B, an anticodon, B, a triplet, or D, an amino acid. 33. The process highlighted in green in the diagram below is called A, transformation, B, X, inactivation, C, translation, or D, transcription. 34. The molecule highlighted in green is called an A, nucleotide, B, amino acid, C, polypeptide/protein, or D, cell. I will double check on your copy of the test for number 34 to make sure that you know the correct answers that are correlated with the correct letter, as they are different on this recorded model. 35. The model or the molecule highlighted in green is called a polypeptide/protein, amino acid, DNA, or mRNA. Similar to the last question, double check to make sure on your paper copy of the test that you have the right correlated letter with the correct answer that you would like to choose for number 34 and 35. 36. A sample of DNA was sequenced and it was found to have 20% of the nitrogenous base adenine. Using Chargaff's rule, what percentage would you expect to have of cytosine? A, 30%, B, 40%, C, 20%, or D, 10%. 37. When one DNA molecule is replicated, the result is two DNA molecules. What is true of the second DNA molecule? The sequence of bases from top to bottom is opposite the sequence in the first DNA molecule. D, it is twice the size of the first DNA molecule. It is half the size of the first DNA molecule, or it is identical or nearly identical to the first DNA molecule. Please double check to make sure you have the correct correlated answer with the correct letter you would like to choose. 38. A blank is a segment or section of DNA that determines the structure of a protein and ultimately a trait. A, gene, B, nucleotide, C, bacteriophage, or D, mitochondria. 39. Which of the following would be complementary to the DNA segment? Would it be AACTAGGC, GGTTGATC, TTGATCCG, CCAGCTTA?